Hello dear students. So far we have seen post-pollination process wherein we have seen how this pollen tube is germinating on the stigma and it is reaching the ovule through the style and the ovary through the to this micropylar end of the ovule. After reaching the micropylar end, we know it is guided by the filiform apparatus which is present in the synergid cells of micropylar end. This is guiding the pollen tube to reach the egg cell. So this pollen tube is carrying the two male gametes and these two male gametes are ready now for the fertilization. So that is what we are going to discuss today about fertilization. So since there are two male gametes, we have to remember them. These two male gametes are having the different fusions. So starting with this, it is called as double fertilization. Double fertilization, we will name why it is called as double fertilization, not just fertilization. So steps in this double fertilization. So pollen tube entering the synergids, pollen tube, this is releasing the two male gametes. So once this pollen tube is reaching the micropylar end of the ovule, it is entering inside the synergid cells through this filiform apparatus. And here this pollen tube is getting ruptured. This pollen tube is getting ruptured and this degeneration of this tube nucleus occurs first and then this pollen tube is rupturing out so that these male gametes are coming out and these two male gametes are ready now for the fertilization. So these two male gametes are coming out and they are getting in the cytoplasm of the synergid cell. From here the two male gametes they are moving individually. So first male gamete is moving towards the egg cell. So we know egg cell is present in the egg apparatus along with the synergid cells. You remember that in the egg apparatus there are three cells, two synergid cell and one egg cell. So this male gamete is moving towards the egg cell and there it is going for the fusion and it fuses with the nucleus of the egg cell. So this fusion of egg cell with this male gamete. So fusion of these two gametes is called as syngamy. So in the syngamy, there is a fusion of male gamete with the egg cell. So this syngamy is also called as fertilization. So fertilization is nothing but fusion of gametes, male and female gametes is called as fertilization. So here this syngamy is leading to formation of zygote. So the formation of zygote is due to fusion of male and female gametes. Male gamete is here the sperm and the female gamete is the egg cell. So this fusion is bringing about the formation of zygote. And remember this zygote is diploid. Diploid is having two sets of chromosomes and these individually gametes are carrying haploid set of chromosomes. So since they are fusing together, it forms a diploid zygote. So this is about one male gamete. What about the other male gamete? The other male gamete is moving towards the polar nuclei. You remember in the these polar nuclei, they are present in the central cell. They are present in the central cell and they are having the two nucleus together. Means these two nuclei are close by and they are not divided separately by a cell. Means they both are present in the same cell, so it is a diploid. And here these two polar nuclei are located in the central cell. So this male gamete is fusing with this polar nuclei to produce a primary endosperm nucleus. So that is primary endosperm nucleus. It is a triploid. It is a triploid nucleus, which means that since these polar nuclei are already having the two nuclei, means they are having individual nucleus. There are two nuclei, and this male gamete is carrying one more nuclei. So fusion of these three nuclei is forming a triploid primary endosperm nucleus. So the formation of nucleus after this fertilization is called as primary endosperm nucleus. In short, it is called as pen. So this pen is a triploid. Triploid means it has three sets of chromosomes. Three sets of chromosomes are there in the pen. So this is very important. Usually they are giving the question like they will ask you to find out the ploidy level of each of these. So they will give you any gamete. The gametes are usually haploid. They will give you zygote, which is diploid. They will give you endosperm, that is triploid. So they will give you this pen primary endosperm nucleus. You know it is triploid because it is fusing. It is forming due to fusion of two polar nuclei with a male gamete. So therefore it is a triploid it is a triploid nucleus. So it is a primary endosperm nucleus, which is a triploid. So this formation of PEN, primary endosperm nucleus, involves the fusion of three haploid nuclei. So three haploid nuclei, they are fusing together, hence it is called as triple fusion. So very important, this triple fusion is leading to primary endosperm nucleus. And here in the zygote, there are only two cells, male gamete and then egg cell, that is zygote. But here this in the polar nuclei, there are already two already there are two nuclei, and to which this male gamete is fusing, so it is a triploid fusion. So triple fusion, sorry. So this triple fusion is leading to formation of primary endosperm nucleus, which is a triploid nucleus. So if you see here, this picture clearly represents the double fertilization. So why we call double fertilization, we are going to just see now. The pollen germination is leading to formation of pollen tube, and this pollen tube is it is reaching the ovule through the style and this ovary and this ovarian cavity, and through this ovarian cavity it is reaching the micropylar end. So here in the micropylar end, uh, this tube nucleus is rupturing out. Before that, there is a tube nucleus which is degenerating, and uh, this pollen tube is rupturing out, so that these two male gametes are discharged outside. And these two male gametes are, one is carrying to the egg cell. So here this is the egg cell, and the neighboring ones are synergid cells. So these all together it is a egg apparatus. So this is an egg apparatus, and it is having two synergid cells and one egg cell. So the first male gamete is fusing with this egg cell to form the fertilization. So this fertilization or the syngamy is leading to formation of zygote. Syngamy is leading to formation of zygote, which is a diploid cell. So later on, the other the other male gamete is fusing with these polar nuclei. These polar nuclei which are present in the central cell. This all together is a central cell. So this central cell is having the two nuclei. These two nuclei are called as polar nuclei. We know why they are called as polar nuclei, right? Because they have migrated from the opposite poles. So they are called as polar nuclei. And these polar nuclei are present in the same cell. So the other male gamete is going to fuse them together, these two polar nuclei, to form a triple fusion. So this triple fusion is leading to formation of primary endosperm nucleus, that is PEN. So this is forming primary endosperm nucleus, that is PEN. So this primary endosperm nucleus is triploid. So this triploid primary endosperm nucleus, we must remember this. So thereafter what happens is this primary endosperm nucleus, it will undergo various divisions to form the endosperm. We are going to see that shortly. So why it is called as double fertilization? Since this angiosperm, the flowering plants, they are showing this fusion. There are two types of fusion shown, syngamy, and they are showing the triple fusion. Syngamy is leading to zygote formation. Triple fusion is leading to formation of primary endosperm nucleus. So since there are two kinds of fusion occurring in the same embryo sac, so it is called as double fertilization. So this double fertilization is exclusively seen only in the angiosperms, which means they are seen only in the flowering plants. So only flowering plants are exhibiting this double fertilization. So only angiosperms, no gymnosperms, no pteridophytes, no bryophytes are exhibiting this kind of double fertilization. So this double fertilization is a characteristic feature of angiosperms. And if you see here in the picture, if you see the embryo sac before fertilization and after fertilization. So after fertilization there are various changes occurring here. So this is remember, this embryo sac is inverted, which means the micropylar end in the previous picture we have seen here, but the micropylar end here it is shown here. And in this synergid cells are degenerating, and this is the egg cell which is developing to form the zygote. So zygote is going to develop into embryo, and in the central cell they are having this primary endosperm nucleus, which is a triploid, and this primary endosperm nucleus is undergoing division further. We are going to see that. On the other side, in the chalazal end, there are three antipodal cells, and these three antipodal cells are degenerating. So therefore, this is how the embryo sac after the fertilization looks like. So even this picture is very important. Usually they might ask this in the exam. They would ask you to draw an embryo sac showing the primary endosperm nucleus, primary endosperm cell, and zygote after the fertilization in the angiosperm. So then you have to go for this diagram showing all these zygote formation, primary endosperm nucleus, and this cell which is having this in the central cell, that is primary endosperm cell. So this double fertilization, we have called that this exclusively seen only in the angiosperms. After the triple fusion, the central cell, so which is around this polar nuclei, the central cell. So we know central cell is largest one. So this central cell is forming into primary endosperm cell. So this is forming into primary endosperm cell. So it is very easy. The polar nuclei is forming to form primary endosperm nucleus, and this central cell is forming to it is forming primary endosperm cell. So PEN PEC. So primary endosperm cell is forming this further later on to the endosperm. So this primary endosperm cell, that is central cell after fertilization, it forms the primary endosperm cell, and this primary endosperm cell is later on after the development it forms the endosperm. So we are going to see what is endosperm, how this endosperm is developing in the post-fertilization events. So on the zygote that is formed after the syngamy, the first fusion, that is syngamy, it forms into the embryo. So zygote is forming into the embryo. So this is embryo development. So this embryo development and the endosperm formation, these are all coming under post fertilization events that we are going to see. So far we have understood what is double fertilization, in which organisms this double fertilization is shown, and why it is called as double fertilization, and what are those two fusions that are occurring there, and what are those two fusions are leading to form. So we have understood this much in the double fertilization, and we have said this is only seen in angiosperms.